ASLO president. And I have to say that I'm extremely excited to be hosting today's event. This is a project that sort of has emerged in ASLO over the last few months that has been finally now taking shape. So the Amplifying Voices series, the purpose of this series is to promote the science of historically excluded groups in our community and provide opportunities for early career researchers who may have restricted funds for travel to network at an ASLO meeting. So the aim through this process is to make ASLO more equitable and inclusive and promote the diverse voices that contribute to our community and to the advancement of aquatic sciences. So the focus is promoting the science. The process is completely open. It's an open call to all members of the aquatic community. You don't even have to be an ASLO member. And the information is available on our website, and we've been promoting it through Twitter among other social media outlets. So it's a wonderful opportunity to promote the science and get to know new people. Let's see, what else did I want to say? The process and many of the creative elements brought to this charge was done by the Early Career Committee in ASLO. So I wish to thank them personally, and I'd like to name the various members because it's a very diverse group of people who have been very devoted to this, creating this series. So I'd like to begin by thanking Hans-Peter Grossart, who's currently the Early Career Committee chair and also member of the board. Thank you, Hans-Peter. Yin Peng Li from the University of Tokyo, Karen Meinekmann from the Julius Kuhn Institute in Germany, Mina Bisik from IGP in Berlin, Germany as well, and Wisha Ghosh, who's at the Indian Institute of Science, Education and Research, Kelly Lewis, who's at the University of Massachusetts in Boston, and Cal Gushlak, who's at the University of Regina in Saskatchewan, Canada. So we have a very diverse early career committee. So I think it's a really wonderful, it's wonderful that they've taken the leadership on this. So I want to begin by introducing our first speaker, and the way in which we will be organized today is our speakers will speak for 20 minutes with 5 minutes of questions. I'll host the questions. The questions can either go in the chat or you can raise your hand. And I will probably invite you to ask your question. And if you feel comfortable, I also invite you to perhaps show your video if you have it, so that it can be a little bit more intimate. So our first speaker is Brittni Bertolet. So Brittni is an Asian American first generation research scholar. And she did her undergraduate studies at the University of Berkeley in 2016 in environmental sciences. And then she did her PhD at the University of Notre Dame, where she studied methane, and she received that in 2021, where she studied methane production in lake sediments across microbial and ecosystem scales. She's currently a postdoc at and an NSF fellow at the University of California, Irvine, which she started this year. And she is investigating community interactions in the scaling of microbial processes. So I invite you all to welcome Brittni to be our very first Amplifying Voices speaker.
Hi, everyone. Thanks so much for having me. I'm going to share my screen now. Oh, wow, what's happening? Is everyone seeing everything okay? I'm going to assume so. Okay, well, thank you so much to Roxanne for the introduction and to the ASLO Early Career Committee for putting on this really awesome series today. I'm really excited to talk about my research, understanding, does microbial diversity really matter? Evidence from lake sediment methane production. So first, I just wanted to start with talking about the importance of microbes in ecosystems. So this is a figure from Ray Lindemann's seminal paper in 1942, often credited with founding the field of ecosystem ecology. And in it, he describes the trophic structure of an aquatic ecosystem, and he puts the bacteria right here in the middle, the bacteria ooze, he calls it. And so back 80 years ago, before sequencing or genomics, even then we knew that microbes were really important and that they were really central and foundational to ecosystem functioning. And then the sequencing revolution happened. And since then, we've learned that microbes are incredibly diverse, both genetically and functionally. But most microbes haven't been cultured. And we know that function is not necessarily taxonomically constrained. So it's really difficult still to understand how the presence or absence of any specific microbial group might be contributing to the larger scale functions that we see in ecosystems and that we care about. And so really the question that drives my research is, how do we use all of this information that we've gathered about the diversity of microorganisms to better understand biological processes and transformations in the environment? So first, why have we ignored microbial diversity for so long? As ecosystem ecologists, we often think about changes in the environment, how that influences a microbial process, and then a specific pool or flux. And I think this framework really comes from early work from a microbial ecology that told us that everything is everywhere, but the environment selects. And because the environment selects, we can kind of just treat microbial ecology as a black box and ignore it, because any of the variation that's existing in these microbial processes is likely captured when we look across differences in the environment. So this is one way that we've been thinking about microbial processes for a really long time now. But obviously, this is beginning to be challenged by a lot of people. There are a lot of papers and really good researchers interested in how variation in microbial community membership and processes influences these microbial processes and has upstream effects on larger scale ecosystem function. And so this is a figure from a really good paper put out by Hall et al. In 2018, where they investigate these questions and really point out the intersection of ecosystem and microbial ecology. But I think there's still something missing here because this still is only reflecting microbial membership as a function of changes in the environment. And what we're beginning to learn from microbial community assembly is that things like dispersal, drift, and even evolution are really important assembly mechanisms that drive microbial membership, and that is not being represented when we only look at differences in the environment. And so one of the things that we're beginning to understand and drives much of my work is that variation in microbial communities can influence ecosystem scale function, and it can do so independent of concurrent changes in the environment. So today I'm hopefully going to convince you that this is true and that microbial diversity really does matter, and doing so with evidence from understanding the role of microbial diversity in lake sediment methane production. So as a quick primer, methane is the global greenhouse gas that contributes to global warming. It has 34 times the warming potential of CO2, and its lakes are an important natural source of methane, and so a really good system for studying the factors that drive methane production and also how microbial diversity influences it. And also what I didn't put on this slide is that methanogenesis is a pretty phylogenetically narrow process, and so is a really good place to start thinking about these links between microbial diversity and ecosystem function. And so just a little bit more on the process of methane production in lakes. Methane is primarily produced in sediments where anoxic conditions promote the anaerobic decomposition of organic material. And this process is really a product of complex combinations of microbial metabolisms and interactions. Methane is only produced or primarily produced by methanogenic archaea here in this black box. And they utilize really simple substrates for their metabolism, things like acetate, hydrogen CO2, and methylated compounds. And so they really rely on other members of the heterotrophic community to break down complex organic material into these simpler components that can be used for growth. And so as we've learned about the diversity of microbes, we're really interested in understanding how composition of these groups influences function. And so for the purpose of today, I'm going to minimize this complexity into two specific functional groups that we'll take a closer look at: the heterotrophs that are responsible for decomposing complex organic material, and then the methanogens themselves, which produce methane as an end product of their metabolism. So as we begin investigating these questions, the first question we asked ourselves was, how does microbial community assembly and function vary across environmental conditions? And to answer this question, we conducted a comparative survey of 14 freshwater lakes at the University of Notre Dame Environmental Research Center. We sampled lakes along gradients of pH and trophic status and measured a number of environmental variables, as well as microbial community composition in the sediment using 16S rRNA sequencing. And what we found was that lake sediment microbial community composition was differentiated across axes of pH and sediment organic matter percent. This is a visualization based on principal coordinate analysis, where every point here represents the community composition of a different lake sample, and the closer they are to each other represent their similarity. Based on this ordination and the distance matrices that go into building this ordination, we can also specifically ask whether different environmental variables are statistically significant in differentiating communities. So here we saw that both pH and organic matter percent were important environmental indicators that correlated with differences in composition across lakes. However, in this study, these variables were not related to methane production. So instead, methane production was best predicted by proxies for organic matter supply, things like total phosphorus and chlorophyll A, and the composition of the heterotrophic community and not the methanogens themselves. And so this led us to really believe that microbial diversity was really important in influencing the composition of the heterotrophs, the ones that are controlling upstream decomposition processes that influences the generation of methanogenesis precursors. And so this was really interesting to us, and one way to answer this question about how microbial diversity and function vary and correlate across the landscape. But it left me a little unsatisfied, particularly with the patterns between microbial community composition, pH, and function, which were all strictly correlated. And so we still really had two driving questions. Is pH a direct selecting mechanism? And we focused on pH because it's one of those variables that often comes out as an important indicator of community composition. We see pH influencing or correlated with community composition across terrestrial environments, across gut microbiomes, and now as well in lakes. So we really wanted to understand whether this was a direct selecting mechanism or simply correlation with other unmeasured variables, or even just due to stochastic processes. Additionally, we really wanted to understand whether this influence of composition on function was a result of microbial diversity, and if it was influencing function independent of changes in the environment. And so first, thinking about this effect of pH, there are a couple of different mechanisms that could lead to this pattern arising in the landscape, where low pH lakes have a specific community composition that differs from high pH lake. It could be that environmental filtering is occurring, in which case different taxa have a competitive ability or a preference that allows them to dominate certain environments over others. Alternatively, dispersal or drift or other Oh, sorry about that. I think, can you guys hear me now? Okay. So alternatively, other stochastic processes like dispersal or drift could be influencing the community and making these patterns arise based on initial assembly conditions or things not related to actual filtering by pH. And so to answer this question, we knew that we wanted to conduct an experiment. And so we conducted a common garden experiment under 2 contrasting pH environments. What this means was that we created 2 separate environments, one with a pH around 4 and one around pH 7.5, in which the environment started as sterile using autoclaved sediment, and then allowing us to really control the pH environment. And then we inoculated these environments with only 1 milligram of sediment from either acidic lakes or alkaline lakes. And we did this in a full factorial design. And so we also included ecological replicates and used 3 different acidic lakes and 3 different alkaline lakes to make sure that we really represented the diversity of communities that exist in the field. We also multiplied this by 4, so we had 4 technical replicates. And then we also included a regional pool inocula. And this was constructed by mixing equal parts of our 6 study lakes. And we did this to really be able to tease apart the influence of environmental filtering if any effect of dispersal limitation was alleviated. So in the regional pool inocula, all of the taxa of all 6 communities had equal opportunity to establish, and so we could rule out dispersal limitation as a mechanism driving assembly in our little incubations. So once these were inoculated, we sealed them, flushed them with nitrogen gas to maintain anoxia, and then incubated them in the dark in the lab for 5 months. And then after 5 months, we measured microbial community composition, as well as CO2 and methane production. And before I get into the results, I want to walk us through a little bit about what we could expect if different mechanisms were important. So again, this is a principal coordinate analysis visualization where the closer 2 points are to each other represent their similarity. So if strong pH filtering is occurring, we would expect communities to cluster by shape instead of color, indicating that pH is really driving differences in community composition, and that the origin of the community where they originally came from isn't really an important mechanism. Alternatively, if strong dispersal limitation is really driving assembly, we would expect communities to cluster based on color instead of pH. And so when we looked at our data, we looked at both the methanogens and the heterotrophs separately, because we knew already that these were really distinct functional components of the microbial community, and that they might have different patterns in how they respond. So for the methanogens, we find that both pH and inoculant source significantly influence community composition. However, we also quantified the effect size of our different treatments, and we found that inoculant source had a much larger effect than pH. And we can see this based on the primary coloring by color, so by inoculant source, and only a secondary coloring by pH. And we're also seeing, pretty a lot of variance between our clusters. We're seeing that really they're not diverging too much. And so there really is the possibility that dispersal limitation and initial assembly conditions matter for driving methanogen community composition. In the heterotrophs, we see a similar pattern in that both pH and innocuous source had significant effects. However, we see a more balanced role of both treatments and tighter clustering within both color and shape. And interestingly, in the heterotrophs specifically, we saw strong divergence across the pH environment in the regional pool inocula, these here in blue. These are not clustering by color at all, indicating that when dispersal limitation was alleviated, pH really was acting as a selecting force and driving these communities to diverge from each other. And so what we're seeing here is evidence that pH does act as an environmental filter on microbial community assembly, particularly on the heterotrophs, but also that the stochastic processes are really important. Dispersal or drift, we can't really rule them out, but we can say that the initial assembly conditions in these communities were really important in driving overall patterns. And so this is really pointing to the importance of considering these different assembly processes in how microbial communities form and how they might be influencing upscale community properties and then microbial processes. So, of course, we also wanted to think about whether this is the case and ask, does community composition influence methane production independent of pH? And because we have this experimental design, we can really specifically test for the independent effects of pH and innocuous source. And what we found is that inoculum source, but not pH, influenced both methane and CO2 production. So communities that originated from near neutral conditions had higher rates of function, irrespective of the pH environment they went into. Additionally, when we look at the regional pool inocula, they were not significantly different from the near neutral conditions, indicating that the regional pool resulted in community rescue of function and equally high rates of both CO2 and methane production. However, there were stronger differences between the pH treatment. And this indicates to us that it's only when dispersal limitation is alleviated and inocula and the communities diverge in this treatment where we saw an effect of the pH environment. So it really is the microbes driving this and the composition of the microbes and not necessarily the environment that they are in. So hopefully I have convinced you that microbial diversity matters and that we should start including it in our understanding of ecosystem function. And so I wanted to leave us with 3 things that I have taken away from this research and also where I hope to see ecosystem ecology and microbial ecology continue to grow in. So first, that variation in microbial communities can influence ecosystem scale function independent of concurrent changes in the environment, and that there really is a utility in utilizing information about microbial community composition in our conceptual and predictive models of function. Second, experimental tests, although they're hard, can really help to disentangle the influence of microbial and environmental drivers separately, but that also our process-based understanding often starts with observation of spatial relationships. But in concert, if we can use comparative surveys with experimental tests, I think it can really allow us to provide robust inferences about how ecosystem responses to environmental change is really a function of biology that occurs across scales. And so lastly, I just wanted to give some hints about what I'm working on now as a postdoc. I'm currently an NSF postdoc fellow at UCI and working to understand the influence of microbial community interactions in the scaling of microbial traits. And I'm doing this now in leaf litter decomposition in grasslands in the context of drought, which is really different from methanogenesis in lakes. But I think that these questions about microbial diversity and function really transcend traditional disciplinary boundaries, and that there's really a unique and important opportunity to share skills, approaches, and understandings as we tackle these questions. So this is really early work right now, but I'm currently using a mathematical model of leaf litter decomposition to ask questions about how community interactions vary under different scenarios of drought and influence larger scale function. From just some initial simulations, we're beginning to understand that community interactions between microbes often lead to higher rates of function than what we would predict from the traits of taxa alone. And so this is really an important component of how we think about diversity function links, especially if the effect of microbial traits are non-additive on larger scale functions. And so hopefully we can apply this understanding of trait variation into our predictions of terrestrial leaf litter decomposition, but also hopefully to better understand variation in stream litter decomposition and potentially also litter fall in coastal ecosystems. And so actually some of these ideas just came out of this year's Eco Days Symposium. And so stay tuned for more work on how we're understanding litter decomposition across the terrestrial aquatic continuum. And with that, I just want to thank my collaborators, particularly those at the University of Notre Dame and the Curie Institute, who really provided so much support for this work, as well as my different funding mechanisms, and I will take any questions.
Thank you, Brittni, for a wonderful talk, very, very clear. And so I'd like to maybe open the floor for some questions. Hans-Peter had one related to your experiment. Hans-Peter, can I invite you to please address it to Brittni?
Yeah, I just want to know if you also took into account two things. So did you look for methanotrophs? Because with the inoculum, you also changed the methanotroph communities. And secondly, did you also take into account that some bacteria also do produce methane in the presence of oxygen, also cyanobacteria and so on? So that would be also related to community composition.
Yeah, I think these are really excellent questions. So we didn't, and primarily because we were focusing on production in sediments and really controlling for anoxic conditions. So we wouldn't expect oxidation of methane to be really occurring. So in the survey, we sampled really deep in the sediments where they're in these like strongly stratified lakes, where any oxidation usually occurs more towards the thermocline. And then in our bottles, we purge those of gas. And so really believe that those are the anoxic conditions were pretty well maintained. But I think that this is a really interesting question, especially as we transfer from thinking about methane production to methane emissions, because obviously, not everything that is produced is going to be emitted. Some of that is going to be consumed by the methanotrophs. As far as the bacteria that produce methane, no, I haven't had a chance to start thinking about that yet. I do think that we should be thinking about it, especially as we try and think about this really in the context of predictive models for ecosystems. But right now, this work was really trying to think about basic questions of assembly and function. And so we kind of tried to really narrow our field of vision into a simpler community that we thought we could answer these questions with. But yeah, as we move to thinking about emissions and really putting this information in predictive models, I absolutely think we have to be considering these other members that are important in methane cycling.
Okay, thank you very much.
Are there any other questions? Please feel free to put it in the chat or to raise your hand. Yes, Jake, please feel free to show yourself if you wish.
I'm here. Okay.
Hi, Jake.
Nice job, Brittni. I was wondering, did you have any time series of the bottle experiments for community composition? Just wondering if there is any, do you have any expectations on how quickly those communities changed, or if you let it go longer, would there be a stronger environmental effect?
Yeah, we really wanted to have a time series effect, but to sample them, we kind of had to destroy the bottles. And so unfortunately, we did have, so this was what we ended up doing is that this 5-month time series was actually our first sampling. We did end up sampling again after 8 months of incubation, but the patterns were just completely different. There was a full shift in community, which I think makes sense. I mean, to sample, you have to completely open the bottles. We also wanted to homogenize it. So we, like shook it up, and that introduced oxygen into the system. And so to use that other time point kind of felt like it would destroy the validity of our experiment. And so we decided just to use the first time point. So yeah, unfortunately, we only have like a beginning and an end point. What I didn't show today is we do have effects, or the communities of just the what we initially inoculated with. So the communities from straight out of the lake. And then we also have like base communities of what could have survived the sterilization process, but we really did convince ourselves that that sterilization process was good. In our control bottles, we didn't see any CO2 or methane production. And so we do also see quite a strong bottle effect, that the communities that are in the bottles are really different from what occurs in a lake ecosystem, which makes some sense to us. Because of what we know about assembly when we change scale and we totally cut off dispersal, and we do all of these other things in the laboratory. So yeah, it's not a full reflection of what we would see at the field scale, but I think an important experimental test of the questions that we're seeing at the field scale.
Cool, thank you.
Julian.
Yeah, thanks. I really enjoyed your talk, Brittni. I had a question that actually kind of piggybacks on what Jake was asking. And I'm just curious, if you ran your bottle experiments a lot longer, do you think that you would get more convergence by pH? Or do you consequently think that they would get more divergent due to different bottle effects or something totally different?
I don't know. I think there's definitely the possibility. I mean, supposedly bacteria turnover times should be relatively quick. And so 5 months of incubation does seem like lots of generations for these. And we really, we only put in a tiny amount of inoculous source. And we didn't measure cell abundances over time, but we did look at changes in DNA abundance, not read counts, but DNA itself. And we did see like a huge 10-fold increase. So like these communities grew up a ton over the 5-month incubation period. So I'm not sure. I think for the methanogens themselves, they're really dispersal limited. So I don't think that we would see a stronger divergence over the pH environment, but maybe for the heterotrophs. And I mean, also like a lot of these patterns is really only explaining like 10-20% of the community, but in a meaningful way that it's still having like direct links to function. So I think that there's definitely going to be change over time, but I'm not sure that we would continue to see changes along the pH gradient. Yeah, it's a good question though. I need to think a little bit more about that too.
Thank you very much, Brittni, and thank you everyone for some really great questions. In the interest of time, I will not ask mine because I think we need to move on to our next speaker. But the one thing I do want to appreciate that Brittni raises, and one of the reasons why I love ASLO is we get to borrow from limnology and oceanography in our work. And so working across different ecosystem types and across different sort of cultural types, particularly at the interface of microbial ecology and ecosystem ecology allows for a much richer landscape. Okay, so I'd like to welcome our next speaker, Patricia Valdespino. So Patricia is Hispanic. And she started with her bachelor's degree in biology at the National Autonomous University of Mexico in Mexico City in 2005. She then did a master's in marine at the Institute of Marine Science and Limnology at the same university in 2008, and then completed her PhD in 2016. She's currently, she then did a postdoctoral fellowship at the Clemente Estable Institute of Biological Research in Uruguay in 2016. And now she is currently a postdoctoral researcher with a Mexis CONACYT fellowship at the Lawrence Berkeley Laboratory, working with their Synchrotron in the US. So Patricia, I invite you to share your screen.
Thank you. Thank you. Thank you, Roxanne. I'm sharing my screen. Please let me know if you can see the full presentation mode. Yeah.
Perfect.
Okay. Well, good morning, America. Good evening, probably Europe. And I'm thrilled to be here. I want to say thank you to the organizers, and also to tell you that it is a wonderful opportunity to share our science with the ASLO community. And my name is Patricia Valdespino, as Roxanne was mentioning. I am a postdoc member of the BSISB program that stands for Berkeley Synchrotron Infrared Structural Biology Imaging Program at the Lawrence Berkeley National Lab. And I will go little by little for you to join me in this exploration of biomineral microenvironments. So when biological systems mediate mineral formation, we call biominerals to these materials. And shells are a good example of this. To the left, you can see calcium carbonate that is a relevant component of shells. And here, the gray dots are calcium atoms that interplay with carbon and oxygen atoms in this really ordered arrangement that repeats because minerals are crystals. And in nature, calcium carbonate can be found with different atomic arrangements, not only this one, that have a particular symmetry, like those you can see below in the left corner. A diversity of microorganisms participate or mediate in biomineral formation. Here I show some examples in the center of prokaryotes, where precipitation can be amorphous, like in the microbial above, or more crystalline in a different prokaryote microbial, where also prokaryotes are main responsibles of this precipitation, more order, more crystalline biominerals. And to the right-hand side, you can see an example of calcifying protists with, you might know that they have a very unique mineral structure. Microbes have conducted the formation of massive carbonate beds in natural waters for billions of years. How massive? Well, as massive that we can see them from space. And for example, here I show the Australian Great Barrier, but also we can talk about the Mesoamerican Reef Barrier in the Caribbean. And stromatolites, similar to the ones you see there in below, constitute the most ancient evidence of life on the planet, dated back as far as 3.4 billions of years. So geologists, like in this photo, geologists have told us and have known from decades ago that microbes are key participants of biomineral formation. However, the microscale of these processes has been an important challenge to understand these processes. We can see mineral now or elemental features with imaging now, for example, with SEM microscopy or with EDS detectors, like those images that you can see to the right-hand side. But we haven't been able to fully integrate the biological information to these processes or to these hypotheses. And this is what took us to synchrotron infrared imaging. And here you see to the left-hand side the advanced light source that is the synchrotron at the Berkeley lab. And briefly, the super bright field beam from synchrotron can be focused into optical systems where samples such as biomineral microenvironments can be studied. Luckily, the main participants in biomineralization, which are biomolecules and minerals, are infrared visible. These two materials have specific features in infrared spectra, as you can see in this spectra I'm showing to the right. This strategy is very generous and is because it is non-destructive and non-invasive. We can explore living cells. Synchrotron has a high signal to noise ratio compared to other benchtop systems, and it can provide chemical information with spatial resolution at microscale. So it is also helpful to study samples with certain kind of roughness, like the samples of environmental biominerals. So in the next slides, I will show you the results from two model systems. The first is microbialites that are the modern analogs or the living analogs of the ancient stromatolites, where only prokaryotes are, or mainly prokaryotes are the participants in biomineral formation. The second model that you can see to the right is very different. And here an eukaryote host and its microbiota are responsible for the formation of the beautiful minerals you can see to the right-hand side. Okay. So we started by making a cross comparison of microbialites from different locations. These are not fossil microbialites. These are living microbial communities actually precipitating biominerals. In aquatic systems of Mexico and Cuba, they are all tropical. In the photos below, you can see the aquatic systems where microbialites develop. And you can see that in general, they are oligotrophic systems with transparent waters that allow a light penetration. These systems have important differences in ionic composition and salinity. And for this study, we integrated the information from synchrotron, FTIR, SEM microscopy, X-ray diffraction, X-ray fluorescence, and 16S metabar coding. And we have been learning a lot about the systems, but in the next slides, I will focus on the results related to synchrotron imaging. So, to start, to the right corner, you can see that in these donut plots, you can see the results of X-ray diffraction about mineral composition. And you can see that calcite, that is the blue, this blue labeled part, it was the most frequent mineral found in the microbialites, and other carbonites such as aragonite in red and hydromagnesite in green were also found, of course, followed by other less abundant minerals. Synchrotron infrared was able to confirm all the minerals detected by X-ray diffraction and provided additional information, such as the hydration state of sulfate minerals, and also something really remarkable that was the presence of organic compounds bonded to silicates. And this was a particularity of karstic environments, and you can see them here in the left-hand side in the infrared spectra shown or highlighted in yellow boxes. And this was a very important feature or very important information to understand the differences of organic carbon content among samples and also of carbon to nitrogen ratios. So what you can see here is the bacalar microbialite, that is the one with the highest content of organic carbon among samples. And you can see that the structure is really amorphous. This is the surface in a SEM micrograph. And at the surface of this microbialite, you can see how a kind of a trabecular polysaccharide or EPS materials are interleaved with areas with amorphous calcites. And synchrotron infrared can detect it right away because these two have very particular infrared features. And here, synchrotron FCIR also provided information that linked structure and function. And here you can see the bright field image of a fresh microbialite surface biofilm on top. And below, you can see the heat maps of extracted infrared signals from exactly the same area. So these are visible up there and infrared below. And here you can see the predominant signal of carbohydrates or EPS in the film. EPS stands for extracellular polymeric substances. And to the right, you can see the spatial signals of proteins and lipids. And to the left, you can see that calcites are located instead in just a few areas. And by overlapping the visible and the infrared, we made a spatial, we were able to do a spatial co-locations of calcites and cyanobacterial filaments that allowed us to link the cyanobacterial polymers and photosynthesis with amorphous calcite formation in the bacalar microbialite. And now we go to the second model system, that is coralline red algae that developed in tidal pools of the Gulf of California near La Paz Bay. And you can see the tidal pools in the top photo. So these are reddish or pinkish colored little trees whose branches are as thick as 1 millimeter, so they are very small. And their aquatic environment is oligotrophic in both summer as winter, as you can see in this box plots. So for this microscale assessment, so we wanted to see the different dimensions of this coralline algae. We integrated again SEM microscopy, X-ray diffraction, elemental analysis, ICP, OES, and metabarcoding and synchrotron FTIR. At the left corner, you can see again the donut plots with the results of X-ray diffraction, showing that magnesium calcite was the most abundant mineral. This is the blue mineral. And we also detected aragonite, another carbonate, and anchorite, that is a mineral in the family of dolomites. So and anchorite was actually more abundant in the summer. And by using Rietveld refinement, we detected the content of magnesium in these calcites that was higher than 18%. And overall, these results just point out that these are minerals with high structural stability. And actually some recent results proved that these minerals can be more resistant to dissolution than other carbonates in decreasing pH conditions, as reported by the group of Nash and collaborators. And well, let's go back to the imaging and to the right-hand side. So I want you to imagine that this mineral environment I'm showing to the right-hand side represents the outer layer or the surface layer of the alga. So the deep layer, as you can see, if we go from the bottom to the top, the deep layer is characterized by a complex structure of thick cell walls, more amorphous in shape. This is called the hypothallus. And above it, and marked in green, you can see the perithallus with a much more defined structure. On top of everything, you see a very thin, kind of flaky layer called the epithallus. And this is what I will show you in more detail in the next slide. So at the center here, you can see the bright field image of a sample of just few tenths of microns of the epithallus that is merged later with the heat maps of infrared signals to the right. So next to this bright field image, you can see the signals of protein and polysaccharides, and you can see that they are actually very low in intensity compared to the high signals of minerals we found in the epithallus. As expected, we found clear signals of magnesium calcite and aragonite, but it was very surprising to find that silicates are important components of this layer. Later, we'll learn that coralline algae are actually able to kind of shed away this layer as a biofouling strategy in a process known as slogging. And well, I came back to this environment or microenvironment because I will show you in the next slides our results on the perithallus, and that we said that it was the growing layer of the algae, these structures that are much more organized. Oh, for this, I want you to imagine that this layer, but from a top view and without the epithallus cover. So here you can see to the left-hand side, to the left corner, that the cells that look more squared in the lateral view are actually hexagonal from the top view. And I will show you next the infrared imaging of this cellular unit, hexagonal unit whose diameter is a bit less than 20 microns. So at the center, below the bright field image, you can see that at this scale, we were at just few tenths of microns, we were able to detect spatial segregation of minerals. The signals of magnesium calcites are more intense to the right, and aragonite signals are actually concentrated to the left. And to the right of these signals, you can see we highlighted on top the position of protein-rich signals. And we found mainly polysaccharides fill the whole cellular content. Something that was very interesting to detect was sulfated polysaccharides concentrated in the cellular interior, and these spatially corresponding with our observations of bulky contents inside the cell. And this was really unexpected because the bulky content of these algal cells has been largely described in the literature as starch granules. We learned that sulfated polysaccharides can be abundant in green and red algae, and they have antimicrobial properties, and they seem to play some roles also in biomineralization. Well, just to wrap up, at this point, you might have some thoughts about the implications of this research. And here I just summarize some of mine. So biominerals have been a relevant component of carbon cycle throughout the history of the planet. The insights of this research can inspire carbon sequestration technologies, which is an urgent need in an increasing CO2 world. This research can help us to understand the stratigraphic record as well, and planet evolution, and also biosignatures of life in other planets. We are learning a lot about microorganisms, structure, ecology, and evolution with these explorations. And these results are also useful to address and model reef and coastal resilience based on the strength or the resistance of their minerals. And more recently, this research is also being used for the development of biomaterials such as biocements. And before getting to the end, and because many ASLO members are marine scientists that might be interested in unicellular protists, in the last slide, I will show some results we just acquired and analyzed from the last beam time that was a few weeks ago, unpublished. Okay, so for the future, we want to move towards microbe chemotyping. So that's complicated because microbes are sometimes really small bacteria in particular. And in this path, we found that this model system, which is a unicellular marine protist from a very unknown clade, if I can say that. Briefly, here you can see that the analysis of 3 different specimens in 3 different rows, and they share morphological features, but also characteristic infrared signals, here named as A, B, C, and D. These chemical signals outstand even if these protists are embedded in a biofilm or sit on top of a biofilm. So in summary, by adding a chemical dimension to microscopy, we can learn about the structure and function of these microorganisms. And these results, or based on these results, we can actually drive our genomic search. And that is really sometimes really helpful. We right now are struggling to determine the chemistry of their wall or shell, if they have a shell. And we are moving forward to determine if this chemical information is species specific or clade specific, maybe. And with this, I finished. This research is possible because of the hard work of enthusiastic groups of Mexico and the United States. Here I show the diverse funding sources. Thank you so much, and I'm happy to take questions.
Thank you so much. I got to get my little clap guy. There you go. Wonderful. So it's so different from what I do. It was really, thank you for taking us on this micro journey, like to the cool biomineralization making mechanisms. It was really awesome. So
thank you, Roxanne.
It was so different. Thank you so much. Hans Peter, I invite you to ask your question if you're still here with us.
Yeah, thank you very much for this exciting talk. I'm just wondering, did you also look, or do you look for the role of cyanobacteria in biomineralization? Because they are also known to drive calcite precipitation in lakes, for example, because they can increase the pH dramatically. I'm just wondering whether you take this into account.
Thank you, Professor Hans Peter. First of all, I just want to say that I'm a huge fan of your science. So I'm very thrilled to be here. And second, I want to say that cyanobacteria are actually our favorite models. And that is because of the size, mainly, because we can study much more features, much more chemical features in these filaments that actually have tens of microns. Instead, working with a small bacteria is much more complicated. And effectively, and there's a long path that has been walked on this path to describe the role of cyanobacteria in biomineral precipitation. Basically, or briefly, the photosynthesis of cyanobacteria creates a partial alkalinization of the surface, and this moves the chemical equilibria towards carbonate precipitation. And if metal ions are available, a calcium carbonate or other carbonate precipitation might occur at the surface or at the complicated surface of cyanobacteria. And we are, this was, what I showed was a pretty basic exploration. And right now we are focusing in disentangle the complicated chemical infrared spectra, because these signals correspond to many molecules that interact in one pixel or one place. So by disentangling or trying to separate the chemical composition of the polysaccharides of cyanobacteria, for example, and exactly where the microsites of mineral precipitation occur, we're going to move forward to get a more detailed map of the role of cyanobacteria in these processes. And one of our systems is very popular. The surface is really populated by Nostocales cyanobacteria with a basal heterocyst. So the conditions also created by nitrogen fixation might be just modifying the outer microenvironment to promote precipitation. So yeah, thank you for the question.
Yeah, very exciting. Thanks a lot.
Thank you.
Very, very interesting. Are there any other questions? I have a small one. I guess it's just, yeah, and I think you partially answered it when you responded to Hans Peter, so I'm sorry if it's a little bit redundant. But in the, when you were looking across different biominerals, or not biominerals, yeah, when you did your study across different microbialites, that was the word I was looking for. Did you see, you know, the notion is trying to look at where the precipitation sites are occurring and what are perhaps the things fueling those precipitation sites, I can imagine, right? And
exactly.
So were different environmental contexts, I mean, getting back a little bit to what Brittni was talking about, could you see drivers of those different environmental contexts maybe influencing what you were seeing, or is it just because I guess the possibility of precipitation is also going to be influenced by the mineral availability in the environment?
Exactly. Thank you for the question. That is a great question. And it is really complicated and that is why we move forward to see into microstructure, because every community has kind of a different arrangement in space, you know? And besides, on top of that, they have different microbial community. So some are more organized and they stratify like in layers, but some are more like networks, like the bacalar microbialite I just showed you, where like different microenvironments, exactly, different microenvironments and gradients of oxygen, gradients of light can be generated at micro scale, right? So that is why we wanted to start like comparing all these sites to see their main differences and their main similarities. But of course, we are moving in towards like getting into the detail of the microenvironments that that explain the microsites of mineral nucleation or mineral formation. So we have learned a lot on the way, and there are important publications relevant to microbialite formation. And in these, in a very broad panorama, I can say that we know that some mechanisms, some microbial metabolisms drive the equilibrium towards carbonate precipitation. And these are basically photosynthesis and anoxygenic photosynthesis and sulfate reduction. Mm-hmm. And these occur only during the day because they, well, not sulfate reduction, but photosynthesis is light dependent. And these create this alkalinization, local alkalinization that promote mineral precipitation. But instead, a big community of heterotrophs as fermenters or different microbes through their metabolism can move the pH, because carbonate formation is pH dependent. So if they have, as Brittni was saying, how important pH is. So if these processes, such as fermentation, move the pH towards more acidic conditions, then a microbial dissolution can occur. And these processes might occur during the day and also at night. So what we see, and the mineral precipitation we see, are the result of this formation dissolution processes that might occur or might change during day and night. So of course, we have detected some of the key participants of these processes. Right now, some more metabolisms are being targeted to be added to the global model, but this is a complicated panorama, as you mentioned.
Fantastic. Thank you very much. Well, it's 10 after the hour. We took a few extra minutes, but we started a little bit late. And I just really want to give a very, very warm thank you to Brittni and to Patricia for 2 excellent talks. And I want to thank the community for attending and participating. I think this was it was really a wonderful, wonderful hour. And I guess with that, I'll welcome everyone to join us for our next Amplifying Voices session on December 9th, where we have 2 speakers from India. And there it is being I'm going to say advertised. So please, if you're interested, there's the website to throw your hat in the ring. This is an open process, and we're really, really excited. We're really excited to have launched this. So thank you very much, Patricia and Brittni, for great, great talks.
Thank you so much.
Yes, thank you.
Bye, everyone. Have a fabulous day.
Thank you. Bye-bye.